Hi, in this video I will discuss a technique known as C100P100 centrifugation assay. So this type of assay is performed to detect whether a protein is present in the membrane or in the cytosolic fraction. So you might be thinking that one protein is inducing other protein to be localized in the membrane. So membrane localization of one protein may be dependent on other protein. So if you ask this type of question, you could perform this kind of assay to find the answer. So the basic principle of the assay is you have to take the tissue lysate or the cell lysate. So in this particular case, you have taken the fly lysate from a Drosophila, and you do the ultracentrifugation. After ultracentrifugation, you would get two type of fraction. One is membrane fraction, another is cytosolic fraction. As the cytosolic fraction is heavier, you would find the cytosolic fraction in the bottom and the membrane fraction at the top. You would differently collect membrane fraction and cytosolic fraction. After collecting membrane fraction and cytosolic fraction, both are individually loaded onto the protein gel. You can see in the protein gel, you would get different bands for membrane bound fraction and for the cytosolic fraction. And after that, you would perform a Western blot and you would use the antibody against B, because you want to see whether protein B is localized in the cytoplasm or B is localized in the membrane. So and whether this localization is dependent upon protein A. So does protein A help protein B to localize into the membrane? This is our question. So you can see there is a Western blot, and in the detailed Western blot you can see there is C, which means a cytoplasmic fraction, M means membrane fraction, and T means total fraction. So in the control condition, you can see the particular protein B is present in both cytoplasm and membrane fraction, but still you can see in the red box, that means in the cytosolic fraction the protein B is mostly present. And when you overexpress protein A, you would see in the yellow box protein B is mainly present in the membrane bound fraction. But see, in the cytoplasmic fraction there is no protein B. That means protein A is actually helping protein B to localize into the membrane. Again, to prove it in the reverse way, we can do a different experiment. Like we can knock down protein A, and in that knockdown condition, we can check the level of protein B. And you can see in this condition, protein B is present in the cytosolic fraction mainly, but not in the membrane fraction. So from these couple of observations, we can say that protein A is actually helping protein B to be localized into the membrane. So this is a utility of C100P100 assay. If you like my video, give it a quick thumbs up. Don't forget to leave comments, and thank you.